Okay guys, this is the third and what I'm hoping is the final video for 9.1. Thanks for hanging with me, and I think you're doing great. And remember to work hard just for me, and I know this sucks, so hang in there. But picking up where we left off, we were talking about xerophytes, and xerophytes are specifically adapted to live in dry conditions. There are other than the desert, there are other environments that have these really challenging environments for plants to have to adapt to. So we know the desert, high temperature, low precipitation. Other areas of the world, things like high altitude and high latitude, typically have really low precipitation. The tundra, where water is typically locked up as snow or ice. It's really sandy soil, which here in Florida especially, many places of Florida have sandy soil, which cause water to rapidly drain. And the last one, which we haven't talked about yet, is areas where the shorelines typically, or the soil, have high salt levels. So all of these plants have to survive in these difficult environments. Specifically, we're going to talk about the adaptations more than just from that first slide, but we're going to go one at a time through the adaptations. Waxy leaves, we've mentioned this a few times, or I have. You haven't said anything. You've been very quiet. You guys really need to wake up and start participating more. I'm just saying. The waxy leaves, the waxy cuticle is going to thicken the epidermis and basically repel water, which means it's going to be trapped inside and you're going to have less evaporation. So, so the thicker the cuticle, the more water retention it's going to have. Another adaptation is found in firs and pines. Particular plants live in really wide ranges of environments, some that have very little water. And because of that, they have these modified leaves, which we talked about already, that essentially have been turned into needles. They've been rolled, and they have a really thick, waxy cuticle, sunken stomata, and no lower epidermis, which allows for them to retain lots of water and live in pretty dry environments. Succulents, my favorite kind of plant because I never have to remember to water them. In fact, when you do water a succulent, more often than not, they just shrivel up and die because they have no idea what to do with the water. Succulents, the leaves are modified again to these needles. This reduces transpiration. The stem, so this whole thing, this is just fleshy. This is actually the stem, and this is where all the water's stored. And this is, the stem has now become the main photosynthetic tissue, whereas in most regular trees and plants, the stem does very little photosynthesis, the leaves do most of it. But since these leaves have been so insanely modified, they cannot do photosynthesis. And therefore, the stem holds all the water and does photosynthesis. Okay, our last adaptation is talking about organisms that live in sandy dunes. So this is where the sand holds no water. It just percolates right through to the groundwater and can't retain it. So these plants, again, you'll see the same theme. They have really thick, waxy epidermises. They have modified leaves. You can see these leaves have been rolled up. The leaves have hair, which help trap the water vapor and slow transpiration. And in this case, the groove formed by the rolled leaf acts as a channel for rain. It's very clever. And the water is going to drain directly to the specific root of the grass and of the stem of the grass. So it's sort of like a channel that collects the water when it rains. Okay, so based on what I just taught you, what we just talked about, can you explain with 8 marks how abiotic factors affect the rate of transpiration in terrestrial plants? Give it a shot. 8 marks. So I want you to check how you did. Read through the options and see where you were missing based on this moderator's note. And I want you to take note. I always tell you that if you are struggling to come up with some answers, if you make a graph or a picture, it's going to give you additional points. Okay, so osmosis is the movement of water across a membrane from high concentrations of water to low concentrations, or from low concentrations of solute to high concentration of solute. And remember, solute is, we typically think of it as salt, but it's really any kind of ion. So what happens is the roots, they will absorb the ions, and there is a high concentration of ions and minerals right here at the base of the root. What this is going to do, this is going to draw the water into the root so that it can then be picked up, swept up, and brought into the xylem vessel. As we know, we need a large surface area for the most transport possible within this cell. In order to do that, we have the root version of a villi, and this is known as a root hair. So everything you know about a villi is very similar to a root, where we have folds that allow for an increase in surface area so we can maximize our rate of osmosis. So what this slide is trying to tell you is basically that in order to get a high amount of ions or minerals into the roots, they have to be actively pumped to get there. Active uptake means that we are pumping a higher concentration of ions into the root hairs so that water will follow. Ways that water is going to move along the stem. And we know that it moves by osmosis, and it follows the solute concentration from low solutes to high solutes. There is symplastic pathway, and that's what you see here in this diagram, where it's going from B to C, where it moves along a solute concentration. And there is these connections, these pores called plasmodesmata in between the cells that allow basically one giant continuous cytoplasm to be working. And then we have apoplastic pathway, and you can see that's right here, D to E, where water is going to move by what's called capillarity through the cellulose walls. And the hydrogen bonding maintains cohesion, which is going to also go through adhesion on the cell fibers, cellulose fibers. So this is the pathways for water movement, either symplastic or apoplastic. Load water into the xylem. We're going to have to pump these ions from the soil into the cells so that water will follow. The concentration of mineral ions in the root is 100 times greater than the water within the soil, because the roots are actively pumping it across the membrane in order to get inside the cells in the roots so that water will follow. Okay. We're looking at chloride. Chloride is actively pumped, creating this pressure, or creating this positive large amount of chloride ions here so that water follows. Now, sometimes plants have a challenge because the ions end up binding with molecules in the soil. So to help compensate for that, they have different adaptations where, for example, here, fungus can sometimes grow on the surface of the roots, sometimes in the cells of the root itself, to overcome these slow-moving ions as they break, because the ions themselves have bonded with different molecules within the soil. Okay, go ahead and answer me this question. See what you guys know. The correct answer is transpiration pull in the xylem. Okay, so this slide is modeling water transport. Actually already performed this experiment here. This application is using models to show water transport in xylem. And when you guys last year, you did a chromatography lab where you got to witness capillary action as the water was being pulled through adhesion and cohesion up the surface of the paper. We can also show it using capillary tubes. And what I'd like to do is show you a couple of videos, maybe just one. And so this is chromatography, which you've already done in class. I'm going to show you at least one video modeling water transport in xylem so that you can at least see a few more real-life model examples. Okay, so I wanted to show you this chromatography video because just to refresh your memory on what we did. Though when we did our chromatography, we didn't use ink, we used chlorophyll from a plant leaf. Check it out.
Then what we want to do is insert that chromatography paper into a beaker with a small volume of water in it. We can see that the water will start to rise up the chromatography paper because of capillary action. You'll notice it's happening very quickly because this is 16 times speed playback. We can see that the ink, which originally looked black, is starting to separate into different colors. This is because the different pigments have different properties. Some of them are heavier, some are lighter, some are more soluble in water, some are less. Because of these different properties, all of the different colors are going to travel at different speeds.
Okay. The last one I'll just show you quickly. One more example of capillary action.
Today we're going to do a cool water experiment. Pretty cool. Cool water experiment. This experiment uses the capillary effect. The capillary effect is the movement of a liquid along the surface of a solid caused by the attraction of molecules of the liquid to the molecules of the solid. Dip one of the ends of paper into the colored water and put the other end into the empty glass beside it. Leave it this way for a few hours. You can observe that the colored water gets transferred into the empty glass through the paper towel.
These are just some quick examples of capillary action and how they work. And we're going to be doing this experiment in class. This is, I've purchased some potometers for you, and you're going to be setting up an experiment to test for different variables. We're going to be looking at specifically what temperature and humidity do. And potometers, they basically are designed to test the rate of transpiration. And as the plant has evaporation from its leaves, as it goes through transpiration, it's actually going to move the bubble towards the tube or towards the plant, and you can measure the rate of transpiration this way. I want you to think about what sorts of factors you're going to need to control, what types of variables you might want to test for, and how reliable your data might be if you only do one repetitious trial. So you're going to be doing this as a class, and more to come on that.